Friends, welcome to Dr. Varsha Gaitonde's channel. In this lecture, we shall talk about systems of mating. In the picture, you may find one very important personality, that is nothing but the scientist Civil Wright in 1921, who contributed for the concept of systems of mating. When we talk about the plant reproduction, there are two kinds. One is sexual reproduction and asexual reproduction. If we exclusively talk about the sexual reproduction, again we divide the plant into self-pollinated, often cross-pollinated and cross-pollinated. As you might have known that self-pollinated crops originated from cross-pollinated crops. Similarly, often cross-pollinated also follow the way of self-pollination. So now we are talking about the systems of mating. System means it's a way how the crossing or selfing is made in a definite fashion. Let us see the definition. Systems of mating is a method by which individuals are paired or assorted for crossing. The another definition is various schemes which are used for crossing or mating of individuals. Here, selfing or crossing, how the different gametic positions or how the reproductive parts are placed on different plant parts or on different plants, like in case of dioecious, or in the same flower condition like in case of rice, how they are originated or how they are placed, that matters a lot. Based on the systems, it is classified into 5 systems like random mating, genetic assortative mating, genetic disortative mating, similarly phenotypic assortative mating and phenotypic disortative mating. As you see, the genetic means the genetic constitution of particular individuals, and phenotypic means the external appearance of that particular organism. Okay, so first one, let us talk about the random mating system. Random mating means, if we talk about the Hardy-Weinberg principle, it talks about the random mating population where the gene and genotypic system remains constant generation of generation, provided there is no selection, mutation, migration or random genetic drift. Likewise, in random mating, what happens here, each female gamete has equal chance to unite with every male gamete. It's a system of outbreeding. There are two kinds of things comes under random mating. One is random mating with selection and random mating without selection. First of all, we shall talk about the factors affecting random mating. Here the factors are difference in flowering time, self incompatibility, male sterility and all these systems. Like when we should talk about the random mating, we don't know whether it is self-pollinated or cross-pollinated. Irrespective of any goal, we are just allowing for open pollination. Here, position of plants in the field, wind direction. In plant breeding, some form of selection is practiced, like mating always practiced. Such a system is called random mating with selection. So we shall talk about random mating with selection. Here what will happen? This will increase the frequency of alleles for which selection is practiced. It reduces the frequency of other alleles. That means, मान लीजिए this is an population and here so many individuals, maybe 1,000 individuals are there. Okay, so what we are doing, we are just selecting these individuals, hardly 50 or so. So what will happen, the selected genes or the alleles which are present in that particular genes are only increased, not all the alleles present in this particular population. So the frequency of other alleles, like this we have left this, all this will be decreased or this won't be there in the selected population. So it will increase the variance and changes mean of character. Random mating with selection increases the frequency of alleles for which selection is practiced and reduces the frequency of other alleles. Here, it alters the gene frequency and population mean but has little effect on homozygosity, population variance and genetic correlation between relatives. The second factor is random mating without selection. Here there is no change, like there is nothing like change in gene frequency, variability, population mean, or genetic correlation between the relatives in the population. So let us see where this random mating is utilized. Random mating is useful in developing and maintaining the synthetic and composite populations. As you might have known that after the development of synthetic and composite populations for 4 to 5 years, to the extent where the farmers use it, they'll use it with the open pollination system. Progeny testing, production and maintenance of synthetics and composite varieties, then production of polycross progenies, evolutionary advantages are the different or various utilities of random mating, where the diversity is maintained to an enormous extent. The second type of mating system is genetic assortative mating. This is again called as inbreeding system. Okay, always remember that genetic assortative mating is also called as inbreeding. Here, mating occurs between individuals that are more closely related by ancestry than in random mating. Like if we talk about the genotype, we shall just cross the two kinds which are genetically very related or genetically very similar. Here the population or effect of inbreeding on population are increased homozygosity and decreased heterozygosity. Here characters or alleles get fixed. Genetic variability of the population is increased, but within the line is reduced. Okay, if we talk about the population means this is a population means there will be several lines. Okay, so in several lines what will happen, within a line the uniformity or homozygosity will increase, so that what will happen, between the lines the variability will increase, so that diversity of this particular population will be maintained to a greater extent. But this is a bane to the line where the homozygosity is happening or inbreeding is happening, because inbreds are maintained through continuous selfing process, so there will be accumulation of deleterious alleles, so that the performance of that particular line will be reduced over a generation of time. The characters of phenotypic assortative mating. Here, without selection, what will happen? The total variability among lines is increased and within line is decreased due to random fixation of genes into different families. With selection, what will happen? Variability is reduced towards the direction of selection. As just to refresh your knowledge, there will be three type of selection process. Okay, one is directional means either this direction or this direction, then disruptive mating, stabilized selection. So when we talk about the directional selection, it goes only in one direction. Disruptive means any direction is possible, and stabilized selection where stability is maintained through the normalized curve. Okay, so when the with selection occurs, variability is reduced towards the direction of selection. That means selection is going either in positive direction or in negative direction, so directional selection is operating here. Here in this case, homozygosity increases due to fixation of genes. Deleterious recessive genes are exposed to natural selection, so that they are eliminated. Then heterozygosity means elimination or decrease in heterozygosity takes place from a population due to fixation of genes. Then heterozygosity is rapidly reduced due to fixation of genes, thus reducing rapidly. Population mean is also reduced due to number of hybrid genotypes which have more number of dominant genes, like capital A small b or capital A small a capital B. Heterozygous condition is exposed to inbreeding, means what will happen, four gametes will be formed or four in four progenies will be resulted, so that here among the four homozygotes obtained from this particular crossing, what we will obtain? We will obtain only one dominant gene which is completely homozygous. Then genetic correlation between relatives is increased due to increase in prepotency. That means prepotency is increased, so that genetic correlation is also increased. So what is prepotency? It is the property of an individual to produce progeny which are identical to each other and as well as to their parents. The utility of genetic assortative mating is for developing the inbreds. Next is genetic disortative mating. This is also called as outcrossing. This is just opposite to genetic assortative mating. Here, genetically dissimilar individuals or less closely related individuals are crossed. So what are the genetic consequences? So variability increases due to combining the genes from two or more different sources and subsequent segregation. Then heterozygosity increases due to combining genes from different lines. Homozygosity reduces obviously. Population mean increases due to heterozygosity. It combines more dominant genes from different lines. Then genetic correlation decreases due to decrease in prepotency and homozygosity. Utility is useful in developing hybrids, polycrosses, etc. Next is phenotypic assortative mating. Here, mating between individuals which are phenotypically more similar than that would be expected under random mating. That means externally or external appearance is quite similar between the individuals which are undergoing crossing. Like here you can see the allelic combinations, the crossing between capital AA and capital AA or capital small a or small aa. Likewise the crossing will be done. Here only two extreme phenotype, that is lowest and highest, remain in the population. Either highest will remain or lowest will remain. So what is the effect on variability? Variability increases since it divides the population into two extreme phenotypes. Then homozygosity, it leads to complete homozygosity in single generation. Genetic correlation, perfect genetic correlation between number of progenies is achieved in one generation. Then increases the prepotency, homozygosity. Population mean will be divided into two groups according to its variability. Then in some breeding schemes, like recurrent selection. This is very important. So where it is utilized in isolating the extreme phenotypes, then changes in mating system disappear randomly when random mating is restored. That means phenotypic assortative mating can be converted, or it can be done to or changed to random mating by a one generation of, or subjecting the population to single generation of random mating. The last one is phenotypic disortative mating. This means mating between phenotypically dissimilar individuals belonging to same population. That is, mating between individuals having genotypes capital AA and small aa. Okay. So in phenotypic assortative mating, what we are crossing, we were just crossing like this kind of allelic combinations. But in disortative mating, what we are doing, we are just going for this type of crossing. Okay, or this type of crossing where they are quite distinct. What will happen to variability here? Variability remains constant since it reduces inbreeding. Heterozygosity remains constant or slightly increased. Population mean, the gene frequency remain constant, thus population mean also remains constant. Genetic correlation decreases due to decrease in prepotency, then prepotency is decreased obviously. So, utility of phenotypic disortative mating is useful in developing more stable genotypes, like increasing the stability. Then useful in disruptive mating program, like in distant hybridization, in making populations stable, like maintaining the variability. Progeny more desirable than the parents, useful when desirable type is an intermediate one and the available parents have the extreme phenotypes. Maintaining variability in relatively smaller population, phenotypic disortative mating is very important. So here you should always keep in your mind that crossing the extreme phenotypes, what will happen? We can maintain the stability. That means we can follow the normalization curve in the population what we are maintaining in a particular location. One is stability. Okay, then what will happen? Distant hybridization, where the parents are taken from different extremities, so that what will happen, better allelic combination will take place. Then maintaining the smaller population, larger population it is not difficult to maintain, but when we will go for smaller population, if inbreeding is happening in that population, means what will happen, that population may go to extinct. To avoid that, phenotypic disortative mating is very important. So let's summarize all the kind of mating systems. First, we will see the crossing or mating type. In random mating, it is by random process. Genetic assortative means mating between genetically similar individuals. Genetic disortative means mating between genetically dissimilar individuals. Phenotypic assortative, where phenotypically similar individuals are crossed so that two extremes are formed. Then phenotypic disortative mating, where better allelic combinations due to mating the dissimilar individuals. The second point is effect on genetic variability. In random mating, the genetic variability, homozygosity, heterozygosity, population mean, genetic correlation, prepotency, all the things remains constant because it follows the Hardy-Weinberg principle, or it is the base for Mendelian population. So random mating is basically utilized where the population has to be maintained indefinitely over a period of time, just like in synthetics and composites. Genetic assortative mating, here what will happen? The effect of genetic variability increases, then homozygosity increases, heterozygosity decreases, population mean decreases due to decrease in heterozygosity, genetic correlation increases, prepotency increases, and utilized for improving or developing the inbred lines. Next is genetic disortative mating. Here variability increases due to segregation because we are crossing the genetically distinct parents. Homozygosity decreases, heterozygosity increases, population mean increases, genetic correlation decreases, prepotency decreases. So it is utilized for developing the interspecific as well as different kinds of hybrids. Next is phenotypic assortative mating. Phenotypic assortative mating, here we don't know the exact genetic constitution. Just based on the phenotypic uniformity or different phenotypic characters, we are selecting the individuals, like alleles are given here, capital A or capital A into small a, like that we are crossing. What will happen? The effect on genetic variability, here variability increases and maintained in the population. Homozygosity increases with a single generation. Heterozygosity decreases. Population mean, that will divide the population into two according to variability. Genetic correlation increases rapidly. Prepotency also increases. Then its utility is to develop the extreme phenotypes. Then phenotypic disordertive mating. Here phenotypically dissimilar individuals are crossed. Variability maintains the variability, that means intermediate types are boosted. Homozygosity will remain constant or it will reduce. Heterozygosity will be constant or increase. Then population mean, here gene frequency and population mean remains constant. Then genetic correlation decreases due to reduced prepotency. Then prepotency is decreased. This is utilized for developing the interspecific hybrids or disruptive matings. Thank you. I hope the information furnished by me will be helpful for you, and if you have any doubt you can put it in the comment box. Thanks for the patience listening and watching.